But before I get into the talk, I want us to spend a minute to remember a very dear colleague of ours, Prof, who passed away earlier this year. Paul was a brilliant scientist, a very fair-minded reviewer. I served with Paul on the hematology study section of the NIH. Among the many accomplishments Paul gave to us in the field, is shown on the cartoon to your right, was Paul's work that first demonstrated that neutrophils adhere to the intima of blood vessels, where they then trap erythrocytes, sickle erythrocytes, to form vaso-occlusive plugs that we all read and talk about. I'm sure some of you have used that cartoon to my right without necessarily knowing that it was Paul's work. And so we miss him dearly, and we hope to learn from his genteel approach to peer review. So thank you for allowing me to share with you a few moments remembering Paul Frenet. So now the task I've been given this afternoon here in Accra, is the genetics of sickle cell disease. And I have put on the far top right the Adinkra symbol called Sankofa. Sankofa in the Akan language in Ghana literally means go back and fetch. And you can see it symbolizes a bird reaching back and grabbing an egg. Most people probably don't know that this symbol is actually not about the past, but it's about the future. It's about the bird picking an egg, which is going to hatch yet another bird. And in the Ghanaian traditional military circles, the idea is to learn from the past to prepare the next war moving forward. So it's a very interesting title, of Future of Sickle Cell Disease. And I want us to look back to learn from the past to advance forward. I want to start in 1977 when the sickle cell mutation was actually sequenced. This is it was actually a paper in JBC by Morata et al. This was the same year that Frederick Sanger, Maxim Gilbert basically demonstrate a very simple methodology of sequencing the DNA. The following year, 1978, is when the first globin switching meeting occurred. And throughout this talk, I would highlight how our focus on understanding the molecular mechanisms that control the switch of gamma to beta globin has really been the epicenter of genetics of sickle cell disease. That same year is when W.Y. Kahn and Dozy discovered the very first human RFLP, restriction fragment length polymorphism. And sickle cell played a key role in that because they discovered individuals with sickle cell disease had a different length polymorphism from those who don't have sickle cell disease. The RFLPs that Kahn and Dozy discovered led to extensive use of RFLPs to study the chromosomes of individual sickle cell disease. And in 1984, there were a pair publication, PNAS, that identified 4 different types of haplotypes. And again, this dominated our study of the genetics of sickle cell disease for several years. We're all familiar with the Human Genome Project, which spanned 1990 to 2003. And this cartoon I'm showing you really was inspired by a very brilliant review published by Francis Collins in Nature at the time, stopping at 2003. Now, 2007 is when Swilia Finn, working at King's College London, maybe she was at Oxford at the time actually, discovered the quantitative trait locus BCL11A, and which really was a follow-up of our efforts to understand globin switching. 2012 is when H3 Africa was launched, and H3 Africa has really been a major landmark in the expanding genomic studies to this part of the world. 2015, 2017, when CRISPR here makes the cut, it actually became the breakthrough technology by science, and Emmanuel Charpentier and Jennifer Doudna got a Nobel Prize in 2020 for this excellent work they did in uncovering the genetic editing technology that, again, is impacting sickle cell disease. 2018 is when Sickle Gen Africa was launched here in Accra, with our collaborators from Tanzania and Nigeria joining us here in Accra. That same year, our very own Charles Rotimi and his colleagues, using more sophisticated methods than RFLPs, discovered that, in fact, the sickle cell mutation may have occurred in a single person, and thus a more sort of a unicentric origin rather than a multicentric origin that we have studied. So I'm using the strand of DNA as a timeline, and we can still talk about how we're going to unwind this to learn more in the future. But in the next 20 minutes, what I would do is pick key moments, key landmarks that I've shown here, a few of them, to expand on how it has really informed our focus on specific areas of sickle cell genetics and how it will inform us moving forward. For those of you who are not familiar with sickle cell disease, I think it's important to run through some very basic molecular genetics. What you see is a karyotype. The genetic lesion is on the short arm of chromosome 11, where the beta globin gene cluster is located. This is the locus control region, which essentially regulates the efficient expression of all the globin genes in a developmental fashion. The mutation that causes sickle cell disease is a simple A to T mutation in position 6 of the beta globin gene. And this leads to the coding of valine instead of glutamic acid in the beta globin polypeptide. That single amino acid substitution is sufficient to change the biophysical properties of hemoglobin S to the extent that when it's deoxygenated, it polymerizes, causes damage to sickle erythrocytes shown here, causing some of them to become sickled. The sickle hemoglobin sticks to the endothelium, it sticks to other cells. This is the cartoon that Prof has really given us to help us explain the primary pathophysiology of sickle cell disease. So the number one cellular abnormality of sickle cell is adhesion of erythrocytes. The second cellular abnormality is that the sickle erythrocyte breaks up very easily, releasing cell-free hemoglobin and heme into the vasculature that causes a lot of organ complications. So these two basic cellular anomalies, adhesion, vaso-occlusion, and hemolysis, contribute to a spectrum of clinical complications that impact virtually every organ system in our body. This leads to severe morbidity and high rate of mortality. So sickle cell patients, here this is data from the United States, the median age of survival for men and women is about 20 years lower than their counterparts. This bottom right column is showing you the impact of fetal hemoglobin. This is a study done by Ofori Acquah, showing that those with high hemoglobin F have a better survival than those that don't. So this is the basic understanding of sickle cell disease. Over the last 45 years, the focus of the genetics of sickle cell disease really has been on understanding how hemoglobin F is switched off and hemoglobin S is switched on. Now that study actually goes further back than 78. This is a picture of Janet Watson. Janet Watson is a pediatrician who worked in New York, and she discovered that babies she was taking care of, who later on developed severe complications of sickle cell disease, when they were babies, when she was looking after them, they were not sick, and their red cells were not sickling as much. So this is the investigator, physician scientist, who made the very first connection between fetal hemoglobin and sickle cell. And this then led to efforts to understand the fetal, the gamma, to the beta switch that essentially has dominated sickle cell disease research over these years. So Janet Watson gets a lot of credit for making that clinical observation, the link between fetal hemoglobin and clinical expression of sickle cell disease. The very first hemoglobin switching meeting took place in 1978. These were powerhouses, Athanasius and George Stamatiopoulos, who actually started work and became, I believe, the first president of the Gene Therapy Society in the United States. I'm showing you here a picture of the globin switching meeting in Oxford in 2014. And I'm showing all of this to highlight the focus over several decades on understanding the mechanisms, the genetics of why hemoglobin F goes down and hemoglobin X or hemoglobin A comes up. Just let a little bit of advert here. The next hemoglobin switching meeting is going to take place in Crete, in Greece, in May of next year. And one of the organizers is Doug Engel, and Doug Engel is very interested. Anyone who has interesting work yet to be published in the globin field who wants to participate in the meeting in Greece, if you just shoot me an email, my email's at the bottom there. I'll just forward your details to Doug, and you can join the group to push forward this frontier of understanding of how hemoglobin F is regulated. The extensive work on BCL11A that Stuart 18 and his group did, Stuart Orkin and his group picked up the baton, and here was a publication in Science showing correction of the sickle cell disease in adult mice by interfering with basically fetal hemoglobin silencing. So BCL11A is a potent inhibitor of gamma globin. It silences gamma globin, and if you basically get rid of it in an erythroid-specific fashion, you can correct sickle cell disease. This is a very nice study. These are transgenic mouse models. So on the left, you're seeing the blood smear of a transgenic mouse that is making human hemoglobin A. This is a litter mate that is making exclusively human hemoglobin S, and you clearly see has the characteristic sickle cells. And here is C11A null mouse, and as you can see just from the pictures, that the sickle cell phenotype, at least in the erythrocyte, completely gone. Building on this work, Stuart Orkin gave a very interesting talk, again, with a future word in here. This is the future of human experimental genetics. This was, I believe, organized by Jackson in 2019, where Stuart talked about harnessing genetics to cure sickle cell disease. This has led to a publication this year of successful treatment, at least hematologically, of 6 patients who have sickle cell disease by giving them a lentivirus that essentially inhibits BCL11A. This was published in New England earlier this year. Just to show you some data at baseline, this is the F cells. F cells are erythrocytes that contain a certain amount of hemoglobin F, not 100%. And so for these 6 individuals who went through this gene therapy protocol, at baseline, you can see the percentage of their erythrocytes that contain hemoglobin F, and then following transduction with this lentivirus that inhibits BCL11 silencing, you can see that the F cell, at least for one, shot nearly to 100%. The follow-up, I believe, is actually now 30 months because this was published in January. 6 patients, and the data on the right is showing the hemoglobin F per F cell consistent with this. So this was very exciting. These studies started, I believe, in 2020, late 2029. But we had a little slight hiccup when another study using a similar retroviral packaging, a few patients that developed complications to do with, I believe, leukemia, and that led that study to be stopped, and the NIH also paused this particular study. But I know that the FDA in the United States has actually lifted the pause on the other study. So hopefully the issues with the lentiviral has been sorted out, and we can get back on track doing this. CRISPR, as I mentioned, really is the premier gene editing technology, and there have been a study. This was also published earlier this year in January in New England. In this instance, 2 patients with thalassemia, one with thalassemia, one with sickle thalassemia. Just to show you the data, patient 1, if we look at the baseline proportion of hemoglobin, we can tell that this individual has beta thalassemia because they make exclusively really hemoglobin A, but the total hemoglobin is about 9 grams per deciliter, so taken enough. Following infusion with CTX001, this is also targeting BCL11A, by the way, by basically knocking it down so it does not inhibit beta gamma globin expression. And you can see in this individual, after 18 months, their total hemoglobin is basically 14. I mean, the anemia has been completely corrected. This patient has sickle beta plus thalassemia, but maybe 12% of hemoglobin A, 9% hemoglobin F, and then after transduction. So both of these scenarios are using myeloablative gene therapy. I'll show you a slide as to how that is done in a second. But clearly, using CRISPR and targeting BCL11A, we now have clear evidence that you can genetically cure sickle cell disease. This is a review in JCI asking, are genetic approaches still needed to cure sickle cell disease? And the answer clearly is yes. The two papers I shared with you both use myeloablative gene therapy, where hematopoietic stem cells are harvested from patients, ex vivo manipulation with a retroviral vector. In both instances, really targeting BCL11A to prevent the downregulation of gamma globin. Again, the benefits of all those years of doing the hemoglobin switching studies. So we have myeloablative gene therapy, then the gene editing with the CASPA, which is the second study I showed you. I'm not familiar with any non-myeloablative gene editing, but this is where we want to go because the myeloablation sometimes can be a problem. So this is great. But you know, in 2018, the Independent, this was published in London, said gene therapy is not available, but could cost millions over a lifetime, save scientists. Now, earlier this year, a group did this analysis, which was published in JAMA, and it's quite staggering. The cost of doing gene therapy, the two studies that described, is actually over $1 million. And so, whereas the technology is there to do this, it's very, very expensive. And this takes me to this slide. What I'm showing now is a cartoon of the prevalence of sickle cell disease in the world. And as you can see, the majority of cases are in Africa and India. This is a projection for 2010. The projection for 2050 is essentially the same. 2050, I think Nigeria becomes the third or the fifth largest country in the world. And so this is where the sickle cell is a major, major problem. And I just showed you the cost of curing the disease using the current technology we have. Now I want to show you the gross domestic product wealth of the world. This is, you know, how rich countries are. And you can see Africa is, you can barely see Africa, right? And so there isn't a lot of money in the parts of the world where the disease is most prevalent. Just to show that to you here. So this is the GDP for Ghana, okay, in 2019. It's around $67 billion. Tanzania, $63 billion. Senegal, $24 billion. This is not a lot of money compared to the GDP in the United States, where it is in the trillion, and a trillion is a billion. And so the major problem now is the curative therapies that we have work, but they're very, very expensive. The WHO recognized the importance of developing other means to combat sickle cell disease when it released this paper in 2006. And it said, concern at the impact of genetic diseases and sickle cell anemia in particular on global mortality. It offered member countries a way to try and control these diseases by proposing 8 recommendations for governments of member states to develop public health approaches that can be potentially less costly and more affordable. Develop medical genetic services, promote relevant, including health counseling, effective international cooperation in combating sickle cell disease, and so on and so forth. To the best of my knowledge, not a single African country has adopted this WHO plan. So whereas the disease is more prevalent here and the cures are astronomical in cost, some of the basic things we can do are really not being done. A major problem is a lot of individual sickle cell disease are not even diagnosed. So when we speak of the genetics, the future genetics of sickle cell disease, even the study that I shared with you, where Khan and Dozie used RFLP to diagnose sickle cell, is not widely available in this part of the world. We did a screen, basically a health screen following this chief when he was celebrating his 20th anniversary 2019. And we screened 418 children. And we discovered 13 of them had sickle cell disease and did not even know they had sickle cell disease, giving us a prevalence of 3.1%. So undiagnosis is a major problem. The other major problem is misinformation and misdiagnosis. This data I'm showing you is of adults who have voluntarily participated in our Sickle Gen Africa study. This is the type of hemoglobin they reported, self-reported in our case report form. You can see there are only 3 types of hemoglobin states. They said they were either SS, SC, or S-beta thal. And when we analyzed the blood samples from this same group of adults, as you can see, we found several other types of hemoglobin. So in fact, 2.5% of these study participants who have been attending the sickle cell clinic didn't really have the right information about that diagnosis. In fact, one of them actually was hemoglobin AA and was attending the clinic. So there's misinformation, misdiagnosis, which is really a major issue. So moving forward, one of the things that we can do is to improve the communication of genetic health information to families, to the general public. We've started to do this. This is a genetic education and counseling for sickle cell conditions, a very short course that we held here a couple of months ago. The course was accredited by the Ghana Psychology Council for 20 continuous professional development points. Because of the nature of the course, we couldn't take a lot of people, but this is the very first time we're doing this, so that the information about genetics, just passing on the information, that people will have the competencies to actually do this well. We're very grateful for the international faculty that participated in this. We had clinical psychologists, genetic counselors. We are very, very grateful to our colleagues in South Africa from Witwatersrand University of Cape Town, where genetic counseling programs are actually running. They joined us through Zoom. Medical geneticist Ambrose Wonka. Some of you are actually listening to this, and I would like to take this opportunity once again to thank you for helping us to essentially do this course and improve the way that genetics is talked about by healthcare professionals. When we asked these participants whether or not the short course met their objective, essentially they all said very good or excellent. When we asked how they will rate the short course, the various components, the course content, the exercise, the role plays, the presentations, all the feedback was good to excellent. These folks graduated, obviously in the COVID era, we're all covering our faces. And here are these guys with their certificates. It was only a 5-day course, so these are not genetic counselors. And we made it very clear to them that having done this short course will not make them genetic counselors, but at least it will improve the way they talk about genetics, the patterns of inheritance. All of them are working with sickle cell families, but they really don't have the right training. What we're trying to do next is to introduce a Master of Science in Genetic Counseling, which will start the early next year. This will be a 2-year course followed by a 1-year internship to train genetic counselors for the first time in sub-Saharan Africa. As I said, there are only 2 programs on the continent, both of them in South Africa. So improving communication of genetic health information is critical. The other critical part in terms of genetics is to understand the genes that modify the disease. Sickle cell disease genomics network of Africa is aimed at doing exactly that. Our goal is to understand how genetics influence the progression of sickle cell disease to inform management and treatment strategies. As much as we support the curative therapeutics, we have to also understand why some people are at risk for some complications and others are not. The Sickle Gen project is really based, not exclusively, but driven by our understanding of the role of hemolysis in organ dysfunction. This is a review I did with my boss, Mike Gladwin, published in Blood. And essentially what our understanding is that when red cells hemolyze and release cell-free hemoglobin, haptoglobin can bind it, but sickle cell patients really don't have a lot of haptoglobin. When hemoglobin is free in circulation, becomes oxidized, it can cause endothelial dysfunction, kidney injury, pulmonary hypertension. Excess cell-free hemoglobin leads to oxidation of the hemoglobin. The heme in hemoglobin becomes free. Free heme and iron can cause oxidative tissue injury, multi-organ damage. We have these proteins that try to block the deleterious effects of cell-free hemoglobin, heme, and iron. And this is really the basis of our work in Sickle Cell Africa. The idea being that extracellular heme, through signaling through TLR4, causes acute organ damage. We published this in JCI in a mouse model, but the model parallels very closely to the rapidly progressive ACS that has been seen in patients. Recently, studying both patients and mice, we demonstrated that excess heme can also cause acute kidney injury in sickle cell disease. Because whereas excess heme in individuals who don't have sickle cell disease, excess heme is picked up by hemopexin, taken to the liver. This is what is supposed to happen. In sickle cell disease, hemopexin is nearly completely gone. So excess heme is picked up instead by alpha-1 microglobulin, taken to the kidney and causing acute kidney injury. So we know these things can be blocked by these hemolysis defense mechanisms. I've already mentioned the proteins that can do this. And so we basically came together as a group in Sickle Cell Africa to try and study a large cohort of patients to understand the genomics that influence the levels of these cytoprotective proteins. In Ghana, the project is primarily a GWAS on that. For those 5 proteins, we are now at the stage where we'll start genotyping using the H3 Africa array. And the second project is a malaria project which we've had to modify because really we're not seeing a lot of sickle cell patients reporting to the clinics with malaria. So we're now focusing on an ex vivo project. I will share a couple of slides on the third project, which is a genomic, an echocardiovascular project, where we are again going to interrogate the SNPs that we identify in project 1 to see whether they will inform us about the echocardiovascular dysfunction that we see in sickle cell disease. This is the organizational, this is basically the study design for Sickle Cell Africa. We are around here. The project started in 2018, as I showed on that first slide. Our target of 7,000 patients, we've literally recruited all of them. I think we're about 50 shy of that. And there's a lot here. I'm not going to go through all of that in the interest of time, but I just want to focus on the 3 research projects, particularly project 3, where we are trying to understand how genetics makes one sickle cell patient at risk of developing pulmonary hypertension and another patient not developing pulmonary hypertension. I just want to, again, emphasize genetic counseling when it comes to giving results back. There will have to be some counseling done so that we just don't hand results back to patients without prior counseling. We're already doing this for some of the data, such as the hemoglobin type. As I showed you, some of our participants don't actually have the right information about the type of sickle cell they have. And so for that, we are giving the results back to them. But once we begin to get genotyping data, that we will not do. If there's a need to do that, it will be done with prior counseling. So just one slide on the echo data. This is very exciting. So this is preliminary data. I believe some of you may have seen this. Kofi Ani, who is a collaborator on this project, presented this in the H3 Africa Forum a couple of days ago. So it's very exciting. I believe this is actually the largest cohort of echo data we have in sickle cell disease, period. The previous study, the Walk Fast study, which was done in the US, I think the number was only about 300 patients. And so you can see the mean age. I really want to talk about the TI jet velocity here. And this is 4.2% of individuals who have TI jet velocity of greater than 3. This is a high risk for pulmonary hypertension and also mortality And so moving forward, we are building a cohort that will allow us to interrogate this phenotype and determine whether or not there are specific genetic signatures that predispose a small proportion of patients to develop PH in sickle cell disease. So let me summarize. I don't know how quickly we're doing on time. If we go back to where I started from, sequencing the gene in 1977 till 2008, now we are in a position to study elements of this in Africa. We're doing this with CRISPR technology, editing the basically BCL11A to stop it from being expressed and hemoglobin F not to be silenced. We're doing this with 6 patients haven't really been hematologically cured of sickle cell disease. So how do we move forward? As we make the turn from here on, we are very hopeful of attempts by Gates Foundation and now with Novartis, recognizing that the cost of the the gene therapeutic products we have, basically it's not feasible. $1 million is a lot of money. And so as we move forward, the expectation is that there'll be cheaper and more accessible gene-based therapeutics that can be afforded by the majority of patients in places like Ghana, where I'm talking to you from, Tanzania, and also parts of India. Very often we don't remember to talk about India, but India has a very large population of sickle cell patients. Brazil, all these places where the disease is prevalent, the current price tag for curing the disease is really beyond it. Moving forward, this will be great, that one will be able to couple newborn screening, which is still a challenge in this part of the world, with autologous therapy once the baby has been diagnosed with sickle cell disease. Again, the information, how the information is relayed is very important. So genetic counseling is going to be very important. So we need a comprehensive approach of prenatal counseling. If a child is at risk or pregnancy is at risk of sickle cell disease, competency in relaying that information. We also, moving forward, some of these ex vivo manipulation. Clearly, laboratories in Africa, India, Brazil will have to be equipped to do this. If we're going to do autologous transplant, somehow we have to have laboratories here that can, at least hematologists as well, that can actually recover hematopoietic stem cells, mobilize these cells, do the ex vivo correction, and then pass it back into babies who may have sickle cell disease. Whilst this is great, there are millions of adults who already have sickle cell disease. So how do we move forward? This is my last slide. And moving forward, it's going to be very important to incorporate data science to improve the phenotyping of sickle cell disease. So deep phenotyping is going to be critical for us to integrate deep phenotyping with the genetics. And all of this has to be done with cross-cutting themes that are focused on these 3 principles, I believe. Community and public engagement regarding new diagnostic and curative therapies. The community has to be engaged. The community has to be told what is happening. Training at-risk populations in Africa and India in all aspects of genetic medicine. And then developing, I can't see the last part because of this. I guess it's cheap and affordable therapeutics, whether it's genomics, epigenetics, and pharmacology. I want to thank you. I'm speaking to you from one of the offices, an office right here, the West African Genetic Medicine Center. Once again, I want to thank the organizers for inviting me to share my perspective on the way moving forward. And what I'm saying is we'll do that